So I'd like to talk a little bit today about hepatitis delta virus, or almost virus, as it turns out, and some modeling work that I've done on it. This isn't my main project for the year here at Nimbus. At the end, I'll talk a little bit about what I'm going to be working on this year primarily. But there's still some interesting questions to be answered about hepatitis delta. I understand it's a pretty mixed audience. There's a couple mathematicians like myself and more biology backgrounds. So there is a good bit of biology and a good bit of math. So hopefully everybody has something to get bored by. So let's go ahead and get started. Start by talking about hepatitis B virus, which is not hepatitis delta. This is a different virus. It's a very prevalent virus. Something like 300 million people are infected with it, at least transiently, every year. And about 20% of the population has had it at one point. So it's very widely distributed, as you can see. This is from 2005, so a little bit old, but very high concentrations in sub-Saharan Africa, in East Asia, and it used to be very prominent in the Mediterranean region, but not so much anymore because a vaccine has been developed for this particular virus. This turns out to be important for our story because hepatitis delta only can exist in the presence of hepatitis B inside of the same host. So the first part of the talk here is going to be about all of the ways in which hepatitis delta virus is weird and unlike every other virus that you've ever heard of, unless possibly you're a plant virologist, in which case viroids will sound like a similar thing. So with hepatitis B, there are a few different things that can happen if you're infected with it. One is that nothing happens, which can happen with any infection if your immune system is doing its job. Or you could get an acute infection that leads to some pretty bad symptoms for a while and then goes away due to an effective immune reaction. Or it could lead to a chronic infection. Which of these is most likely depends on whether you're a child or an adult. In children, it's much more likely that you get a chronic infection. Something like 90% of cases that are acutely infected become chronic in children, whereas in adults, that's reversed. It's something more like 10 to 20% become chronic infections, and the rest are acute. In an acute infection, you get a high viral load, up to 10 to the 10 copies of this HBV DNA, the DNA of the virus, per milliliter of blood. Lasts for a few weeks, and then it goes away. And it's resolved in most adult cases. I'm going to only be dealing really with adult cases in this, because the immune system is basically completely different for children. And that's a dynamic timescale kind of thing I don't really want to get involved in at this point. In chronic infection, you end up with a lifelong infection. And you have, because of this lifelong infection, maybe I should start with the beginning, right? It's the liver that's being affected. It's hepatitis, right? So hepatocytes are being infected. So in the long term, there's a risk of cirrhosis, increased risk of cirrhosis, and of hepatocellular carcinoma in the long term. So that's basically what it ends up getting you in the end. The mechanism that connects the infection with these symptoms is not entirely clear, as I'll talk about a little bit more with hepatitis delta. Right, so that's hepatitis B. Right? It does the normal virus thing. It infects the cell. It gets in. It's a DNA virus, so what it'll do is it'll get its DNA into the nucleus and then has this circular DNA that will then transcribe into the various proteins, and then it's an enveloped virus. It'll break off a bit of the cell surface and go off into the world to try and infect something else. Right. Now what about hepatitis delta? So this is another hepatitis virus. But it is, instead of the most common form of viral hepatitis, like hepatitis B, it is the least common form of viral hepatitis. But it also has the worst outcomes. And it has the following qualities, which all make it very unusual for a human virus. It has a negative sense circular RNA genome that is extremely short, 1,700 base pairs in length. It only encodes for one protein, one. Right? So it's got this entire genome that's entirely dedicated to making copies of itself and one protein, which has two forms. There's a mutation that occurs to allow a longer form of this protein to be made. I guess if you want to be nitpicky about it, you could say this is having two proteins. Right? You have a long form and a short form. They do different things, but basically it's just one open reading frame that makes this one protein. So for a virus, this is highly unusual. Hepatitis B has tens of proteins that it produces. I don't know the exact number. Most viruses have the ability to produce a fairly large number of different proteins that are specific for its replication cycle inside of the cell. So they don't produce them on their own, obviously. They hijack some of the cellular machinery to make these proteins, but hepatitis delta only has this one. And the really strange thing is that left to its own devices, hepatitis delta can infect a hepatocyte. It can very nicely copy its it does some weird things to copy its RNA genome into an RNA antigenome, and then from that antigenome template to make thousands of copies of its own genome again. So it can fill up the cell with copies of its own genome, but it can't get out. It can't get out of the cell. It's just got some proteins and some RNA sitting around in the cell. It can't get out, unless that cell also happens to be infected with hepatitis B. Because hepatitis delta needs the surface proteins from hepatitis B in order to get out of the cell, to form and envelope for itself and to get out into the world and infect new cells. So it requires in the individual cell a co-infection with this other virus, hepatitis B, which luckily for hepatitis delta is pretty common. Right. So like I was saying, this is just a schematic of what hepatitis delta does. It has a pretty simple life cycle. It's basically every important event in the replication of its genome and making of proteins that happens. And then the last step down here is that it needs hepatitis B surface antigen to make the surface for the new virus and get out of the cell. So there are a lot. So this leads to some strange interactions. So the first question you might ask, that I haven't really addressed in the slide, is where did this thing come from? Where did hepatitis delta come from? And the obvious answer would be, oh, this is just a defective version of hepatitis B. Right? Hepatitis B was going along, and then some kind of freeloader version, like reduced version, figured out, oh, I don't really need to do all of this stuff. If there's already an infection in the cell, I only need to do this minimal amount of things to make a new copy and get out into the world. So defective virus is one possibility. But the fact that hepatitis B is a DNA virus and hepatitis delta is an RNA virus pretty much rules that right out. Right? So somehow this emerged from two independent things that one became a satellite of the other in the long run. So now what we're left with is this system where we have a virus that's perfectly happy on its own and then another one that's freeloading off of it, and they have these dynamical interactions between each other. So for example, one of the two versions of this protein that hepatitis delta produces suppresses the replication of HBV DNA. So that sets up a bit of a game, right? If it's really good at suppressing HBV, it's going to basically destroy the field for itself, right? It's not going to have enough places to infect to then propagate itself, because it needs HBV to survive, but inside of an individual cell, it needs to get those resources directed over toward itself rather than going toward producing more hepatitis B. So there's a bit of a game there where it has to decide how well to do this. So there's some evolutionary pressure in two directions there. And then on a more systemic level, what happens when you have both of these viruses interacting in one individual, infecting the same cell populations? Some strange things can happen. So first of all, there's two ways that you can get both of these viruses. One is that you can get hepatitis B first, have an established chronic infection, and then add in hepatitis delta from a source who also has both. So that's a super infection. And the other possibility is you get them both at the same time. Right? And you can try and think about what might happen in those cases. And it turns out that in the case of co-infection, when you get both of them at the same time, this leads to chronic infection with both of these diseases only 2% of the time, whereas with hepatitis B, it leads to a co-infection a chronic infection 5 to 10% of the time. So getting the co-infection with both of them decreases the risk of getting a chronic infection with either of them, but in particular with B alone. It's a lower chance of getting that chronic infection. In the case of superinfection, where you already have a chronic HBV infection, there's a 90% chance that a superinfection of hepatitis delta will establish on top of that. Basically, the field is ripe for this new virus to come in. It has all the resources it needs. It has lots of cells that it can infect and happily go about its way. So it establishes very easily in those cases. So there's a difference between co-infection and superinfection. But it's not good. But one of the strange things is sometimes when you have a chronic hepatitis B infection and the patient is then super infected with hepatitis delta, both of the infections clear. Right? So somehow adding this extra thing leads to the clearance of something which had been chronically established before. Happens only 1% to 2% of the time. But initial thought is, hey, great, let's take all of our people who have hepatitis B and let's just give them all hepatitis delta too, and then 2% of them will be cured. This is great medicine, right? Except that the other 98% will be in very, very bad trouble because hepatitis delta is not your friend at all. It's a quite bad disease. So it has the same kind of outcomes as hepatitis B. It has increased risk of hepatocellular carcinoma, increased cirrhosis, but much faster and worse than in that case. So in chronic hepatitis delta, you get cirrhosis in 70% of cases, and in some of these cases, in about 15% of the cases, happens in a year or two. That is a very, very fast progression to cirrhosis. You would have to do some serious drinking every morning, noon, and night, I think, to get to this kind of cirrhosis in that kind of speed. It also increases the chances of hepatocellular carcinoma and makes these things, it makes it the onset sooner. Has 10 times the mortality rate of hepatitis B alone. But, and this is where we're going to be getting into the immune system, which is the thing I'd like to focus on from a modeling point of view, neither of these is believed to be directly cytopathic. So neither of these infections is believed to directly harm the hepatocytes that are being infected. So they can kind of happily go along, maybe not happily, but they can go along being infected for quite some time with this virus inside of them. It's not the kind of virus that kind of bursts the cells that they're infecting. It doesn't just damage them in any way that's direct. There's some controversy about this. It isn't entirely clear whether these cells are directly harmed or not, or maybe there's something more indirect, like impaired function of the liver, that kind of thing could be going on here. But they're not believed to be directly cytopathic, so that's going to cause some, that's going to come into the models when we're getting there. Okay, and then just a little bit of biology to go here. As far as prevention and treatment goes, there's a very effective vaccine for hepatitis B. I'm no doctor, but I would say, having read enough papers about these viruses, get this vaccine. This seems like a pretty good idea. Consult your physician, of course, but seems like something that you'd want to do. So if you get this vaccine against HBV, and it works, then you don't get hepatitis B, so then you can't get hepatitis delta, or at least you can't get any kind of sustained hepatitis delta infection. There's no treatment for the acute hepatitis B infections, but acute ones clear themselves. The chronic ones there are treatments for, and they're pretty effective, so they can often lead to clearance. And there's no specific antiviral treatment for hepatitis delta. There are some interferon alpha treatments is the only thing that's approved. It's pretty vague. Nobody really quite knows how it works, what its mechanism of action is. It sometimes does something for some patients, right? But that's about all there is as far as treatment of hepatitis delta goes. And once you have hepatitis B, chronic hepatitis B, there's no way to prevent hepatitis delta infection from establishing, or to try and decrease the chances of it establishing a chronic infection. And that kind of makes sense. I mean, this is a very, very simple virus, so there aren't a lot of targets that you could go after. There are some treatments that are in trial now. I can talk a little bit about that later if we want to. Good. So strange features, right? HBV delta depends on B, but suppresses its replication, so either directly or indirectly. You have this effective treatment for hepatitis B, so you would think, okay, I have B, and then I have delta that depends on B. So if I treat the underlying infection, I should be able to deal with the co-infection as well. It turns out that if you take, at least for some of these treatments, so for example, lamivudine, if you take a treatment that does work against chronic hepatitis B and you give it to a co-infected patient, it basically does nothing. That's something that I'd like to see if I can get a model to reproduce that kind of behavior, right? Right. I mentioned that superinfection with hepatitis delta can lead to resolution of chronic HBV, which is a bit weird, and and it's not known why hepatitis delta leads to worse outcomes, right? That neither of these is directly affecting the cells that are being infected, so something more systemic has to be going on to explain why it's so much worse with this second infection on top, right? All right, and there's also some weird dynamics that's been observed. So a paper in 2010 observed that you have these unexplained fluctuations in the levels of hepatitis B and hepatitis delta that are happening in a single patient. So the thing to pay attention to here are, strangely, the two purple lines, one slightly darker than the other. I am not responsible for this figure. I only use it every time I talk about this topic. So if you pay attention to the dark purple line and the light purple line, the dark is B and the light is delta. And so one possible course is that you have very low levels or undetectable levels of hepatitis B all the time. So that's just flatlined at the bottom there. And then the hepatitis delta levels fluctuate. That's not so unusual that that would happen, since you are suppressing the B replication, and then that could lead to kind of declines in delta replication as it runs out of places to effectively infect, and then comes back as it kind of cycles. You also have some observed fluctuations out of phase with each other. So B will go up, D will go down, and then it'll recover. So it's almost like a predator-prey kind of cycle going on there. And then the strangest one is when you have parallel fluctuations. You'll sometimes see them both go up and down in parallel with each other. So if some of these fluctuations can be encapsulated by modeling, that would be something that that might be a clue that the model that we have is actually doing some of the right things, or including some of the mechanisms that are important for the overall dynamics of the system. All right, so let's try some modeling. What I'd like to do is, what I was aiming to do with this initial work was to develop a model, as simple as I could, that would have all of the necessary compartments of the infected cells with the different types of viruses, and that would be able to reproduce some of this behavior. So, for example, the fluctuations would be great, but mainly that you could come up with some kind of explanation of why it might be that treatment that's effective for a single infection might not be effective for a co-infection with the two different kinds, right? So basic system is you have your hepatocytes. They're uninfected. They can be infected either with one or the other type of virus, so B and D, and then some of them are going to be then co-infected with both. The co-infected cells can produce B virions, not very many of them because of the suppression by delta, and lots of delta virions. Right? And the ones that are infected with B only can produce B virions. The ones infected only with hepatitis delta can do nothing, because we've already said that it's a dependent satellite virus. It needs this co-infection in order to produce anything. So they have to wait until they get into the co-infected class. OK, so a simple model of this. So here's where the fun begins for our non-mathematician friends. But you're in Nimbus, so must be used to seeing this kind of stuff. I don't know. So basic equations. I have hepatocytes that are expanding on a logistic type of dynamics, which makes sense. The liver has a very good ability to regenerate itself. So we're assuming that if it's damaged, it's going to kind of go back to its carrying capacity, its normal size, over a reasonable time course. So there's a logistic growth there, maximum carrying capacity that's shared by all of them. Right? So you have a total number of hepatocytes that's going to stay more or less constant, barring some massive damage that's happening to these cells. And then just some basic passage, just infection of one cell type. I've removed the virus equations by assuming that the viral dynamic timescale is much faster than the timescale for the cells. So you can basically assume that the virus population is in quasi-steady state with the infected cell population, so you can just do everything in terms of equations with just the cell population. So it looks like the cells are infecting each other, but in fact there's this virus, intermediate. Although cell-to-cell, cell-cell infection is also possible. Right. So basically going from uninfected to the different classes. This is just basically what I had on the diagram there. If you do that, then your simulations do basically what you would expect them to do. Right. If you have a you infect with, in the first graph here, you infect with B first, the B infection gets really large, and then you co-infect with Delta at day 300, and then that takes over, and basically everything's co-infected. There's some question about whether this is what's really happening, whether all of the cells are being infected or large numbers. In animal models, that seems to be the case, but then for some other forms of hepatitis, it seems like maybe only 30% of the cells are being infected. That adds a whole new wrinkle to things that I'm not dealing with here, but that's an interesting bit. Also, as far as animal models for this go, not easy, because you need something that first can be infected with something like hepatitis B, and then can also be infected with something like hepatitis delta. So it turns out that this is so far done in woodchucks and some ducks and monkeys. That's about it. None of these are very easy to work with, although there is a group at Georgetown that's actually doing the woodchucks, which has to be an interesting lab. You know. Right, so anyway, this is all pretty basic. There's nothing interesting happening here, except that basically, this is clearly not going to tell you everything, because everything gets infected and that's it. Nothing happens after that. You can give as many drugs as you want. Everybody just stays infected. Nothing happens. So we need to add in a little bit more. Right, so I already talked about this. So this drug that I'm going to be talking about, lamivudine, reduces genome replication of HBV and the number of infected hepatocytes in chronic Hep B. And we're going to see, what I want to see is that the model will do that. If you add a drug, you put in you change the parameters in such a way that it would indicate reduced replication of the virus, then you should see in infection with hepatitis B alone, the amount of infected cells should go down, but in the co-infection, it shouldn't go down. Right? So if you don't have any immune response, this is what happens when you do drug treatment. Nothing. Right? It doesn't do anything. Everything still gets infected. It just maybe takes a little while longer. So we're going to add in a bit of an immune response, because clearly something has to be going on besides just a bunch of cells that are sitting around being infected by stuff. Right? Your body does care, anthropomorphizing a bit here. Your body does care about its liver. Liver is a pretty important organ, and it's huge. So you're dedicating a lot of resources to keeping this thing. It's got to be doing something important. So, and there's a lot of immune cells available at that point, because there's a lot of damage that happens in its normal course of action. It's doing all this filtering of toxins and things like this. So, right, so there's a fair bit that's known about the immune responses that are involved in dealing with hepatitis B, but not so much specifically about what happens with hepatitis delta. So we're going to try and use the kind of dynamical picture that we have to piece together what might be going on with the immune system in response to these two infections happening at the same time. All right, so sorry about the change of color scheme. It's going to get dark here, but I had a bunch of equations that were written in white, and I couldn't put them on those slides. So anyway, here they are. So these are the same equations in white that we had before, and now just adding in some, in red here we have an effector T cell class that's going to be reacting specifically to hepatitis B. You can assume that there's nothing that's specific for hepatitis delta for now, and see if we can just do it with the one type of reaction. If that explains everything, then there's no evidence for or against there being a separate response to hepatitis delta. So source, it's reacting to all of the cells that are infected with either B or with both, because I'm going to assume that, these are the cells that have Hep B in them. So whatever it is that Hep B is doing to elicit this immune response, it's going to be detected on the surface of these cells. And it turns out that the effective responses to hepatitis B are to core proteins. So they're the kind of things that would be present in B but not in delta. Right, so, and then so these are elicited in response to these infected cells. They decay at some rate, and then their effect, at least in this first step of the modeling, is to basically cure the cells of the hepatitis B. So I'm not having them kill the cells and then have them replaced with uninfected ones. Rather, they're going to be cured of this hepatitis B infection. And one of the reasons to do this is that if you assume that you have to kill all of the infected cells in order to clear this viral infection, well, there's an awful lot of cells in the liver. And you would have to, just kind of figuring it out mathematically, you'd have to basically kill every cell in the liver 7 times before you could clear this thing in a dynamic way, right? As with things being reinfected as you go, things like that. There's no evidence that much cell death is happening. Right? That's the kind of thing that you would presumably be able to detect. So in order for this to work on the kind of timescales that we see curing happening in drug treatment or in clearance of acute infections, you have to assume that there's some non-cytolytic way that the immune system manages to get rid of these infected cells. Right? So that's what we're assuming here, that B-infected cells are going to uninfected, and ones that are co-infected, they're clearing the B infection, but they're still infected with Delta. Right? So you might imagine that somehow they're signaling the cells to get rid of this foreign DNA. B-infected cells tend to have somewhere between 1 and 50 copies of the infectious DNA in them. So it's not such a huge stretch of the imagination to think that this could be done. Right? Whereas with hepatitis Delta, there tend to be something on the order of like 10 to 100,000 copies of the genome in each infected cell. So that's a little harder to imagine that being cleared kind of one at a time. Right, so let's see what happens with this model. Well, so do the same things as before, simulate infection. And then on day 500 here, I'm changing the parameters to indicate that the production level of hepatitis B is going down. And you get what you want. The number of infected cells goes down very rapidly. There's a two-phase decline. And the number of uninfected cells recovers. And over the course of time, you essentially clear this virus under this treatment. Great. So let's see what happens with the co-infection. What you don't want to happen, it also works, right? Which, well, you do want to happen, but it's not what really happens. So again, now the co-infection goes away, and you get a population of cells that is just singly infected with hepatitis Delta. Right? That recovers. So this was good and not so good. So adding the immune reaction to hepatitis B explains, or at least recapitulates, the clearance in a drug treatment for a single infection, but it doesn't get the fact that you don't have a clearance in the co-infection. So still need a little bit more work. So what I decided to do is say, OK, well, let's say that there are two separate immune reactions that are happening specific for hepatitis Delta, specific for hepatitis B. They're happening at the same time, again, stimulated by the cells that are infected by that particular type, and then acting against, suppressing those infections. And then also not shown is a competition directly between these two, some kind of resource competition, or indirect, I should say, resource competition between these two effector classes for stimulation. It could be, some kind of co-stimulatory cytokines that are available, right? So there's some kind of competition. You can't just have each of them going whole hog independently and see what happens with this. So the equations change a little bit. We have two different types of effectors in there, obviously. And then for the effector cells, we have the same kind of equations as before, except now with a competition, simple mass action competition term thrown in. And we'll see what happens with that. And it turns out that if you do that, it does work. When you do the treatment in a single infection, it goes away. And when you do the treatment in a co-infection, nothing happens. Right? So you can kind of work through the steps of why that might be, right? Because the hepatitis Delta is kind of taking over the system, you have a strong hepatitis Delta reaction and a low hepatitis B reaction. When you give the drugs, you still have the same thing. So there's not much work going on curing the hepatitis B, because that reaction has been suppressed by the strong reaction to hepatitis Delta. So basically, you get this situation where, because the immune reaction to the primary infection has been suppressed in favor of the dominant secondary infection, you don't get the drugs working because they're relying on this clearance by the immune system happening at the same time. And if you don't include the competition between those two things, then it goes a little bit haywire, and you get clearance in the co-infection system. So, right. So that's the story on that stuff. I got some more stuff to talk about with hepatitis Delta, though. So let me pause here and ask if anybody has any comments or questions about what I've said so far. Yeah, so I'm showing some. Yeah, so analytically, there is a fair bit that you can show about convergence of these things, so or stability. So I haven't gotten any global results, but the local asymptotics are kind of what you would expect for these things. So, yes, that's right. Yeah. Depending on the drug effectiveness, you can estimate things like that. Yeah.
On the slide that you showed with the two purples.
The
two purple graphs.
Oh, yeah.
The antibody response looked very interesting.
Ah.
Now it looked like it was a surface antibody rather than a core antibody.
That's right. Yeah. Yeah. That's surface. Yeah.
I noticed that where the viral loads seem to be equal.
Yeah.
You had a high level
all the way throughout.
Surface antigen.
Yeah.
And wondered if that denotes anything as far as immune response. And if you've looked at core antigen response.
I have not looked at measuring the actual antigen levels. So surface antigen has been something that people have been interested in using because there's tons of it, right? So you can detect it very easily. If you could get some kind of signal out of that to tell you about what the underlying system is doing, especially in these situations where hepatitis B is suppressed to such a low level that it's very difficult, at least with kind of standard tests, as opposed to like more sensitive laboratory tests, to detect whether it's present or not, or to get any kind of levels from that. So, but statistics-wise, they just haven't found that you can get much information from the hepatitis B surface antigen. It's basically flooding the system. I mean, you do see these fluctuations here, or some interesting things happening with those there. But as far as I know, nobody's been able to derive any kind of information about the underlying system based on that particular thing. Yeah. One other thing to note about this is that the time points are really far apart. These are like 6 months, 6-month intervals between data points. It would be really nice to have some dynamics on a little bit smaller time scale. So, who knows how much of this is really regular. I mean, he says fluctuations, but it could just be a question of when you're sampling. Maybe they're all representing the same thing. It just happens to catch it at the right time points for some patients and not for others. Maybe it's always just, going up and down. Okay, so story so far, lamivudine treatment for chronic Hep B and D have very different outcomes, and if you consider two separate immune reactions that are competing with each other for a common resource, then you can recapitulate this. You have to have, so there was a long process and more models than I've shown of kind of model selection, trying to get all of the necessary features and determining what had to be included, what was neither here nor there if you included it or not. And it turns out that having this homeostatic proliferation of the hepatocytes is important. You have to have that kind of underlying mechanism going on, and that this competition between B and D specific CTL responses was necessary to get this to happen, unless there's something completely separate that's explaining it. But it seems like thinking that the immune reactions to the two different infections might be involved in this explanation is the parsimonious thing to say about it. Right? Certainly you have immune reactions. It's just a question of how they're interacting with each other. Okay, so, right, been there. Right, and so, and so the same kind of model with, basically the same model, you can get it also to do some of these fluctuations. You can't get so these are parallel fluctuations, I would say. I'm not sure actually which kind of fluctuations I would call this, but you can get periodic behavior from this model by choosing the parameters in the correct way. It turns out if you have a pretty high competition level between the two immune responses, then you can get into this kind of cycling where they kind of suppress each other one after another. But you can't get all of the different dynamics of those fluctuations. You can't get both the parallel and the out of phase fluctuations this way. So again, you can prove existence and convergence of these kinds of periodic solutions. Okay, so another aspect of hepatitis infection that I've been looking at is what it is that causes the damage to the host, right? If it's not the virus directly killing the cells, damaging the cells, and then needing to be replaced, what is it that's doing the harm? And in cases like this where you have a non-cytopathic infection and you want to know what could possibly be causing these negative outcomes, the answer is usually the immune system, right? When you get a cold, you get something like that, most of the symptoms that you experience are your immune reaction to the infection, not the infection directly causing you to sneeze or cough or have a running nose. Right? You're doing that to try to clear yourself of the virus. So suspect that the immune system is involved in the damage that's being done, especially in this co-infection with hepatitis delta, why it might be worse. And one possible explanation for this comes from the phenomenon of immune exhaustion, which is the stepwise loss of effector function in when you have a prolonged exposure to antigens. So if you have a chronic infection, over the course of time, the immune reaction that you're generating against that infection loses some of its abilities. Right? The cells that are involved will start, stepwise lose their ability to produce different cytokines that are important for that reaction. Basically, it's almost like they're giving up. Right? Well, this isn't working. It's probably causing harm to your host tissue. So they just kind of say, OK, forget it. We're not going to do this forever because maybe we'll die or whatever, right? So CTLs will lose the ability first to produce IL-2, then tumor necrosis factor alpha, and then eventually, sometimes, interferon gamma over the course of a chronic hepatitis B infection. So the thought that I had was that what could be happening to cause the damage in hepatitis delta to be so severe in these chronic cases is that you have a chronic infection with hepatitis B. You generate an immune response, which eventually becomes exhausted. So it's still there, but it's not doing as much damage over the long run as it had been during the acute phase. Now hepatitis delta comes in, and it suppresses the initial infection. Once that initial infection has been suppressed, there's a less stimulus to exhaust the Hep B-specific immune response, which then resumes. Right? And it's now not so stimulated that it has an exhausted response, has a full-on response. There's still tons of infected cells, all of them, basically. And so you could see that this would lead to a chronic, acute-type immune reaction, which would cause more damage over the long run. So I wanted to, and work with a couple of my undergraduate students, we took some models for immune exhaustions and then adapted them to this co-infection type of situation to see what would be predicted by kind of a simple model of what was going on with immune exhaustion if you could explain the symptoms that are being observed in these patients. So here's where we wound up. We started with the model that was originally developed for vaccination. But this is the basic idea. It's a pretty heuristic model. You have uninfected cells that become infected. And the presence of these infected cells leads to two things. One is that it elicits an effector T cell response, so an immune response, which then suppresses these guys. And the other thing that the presence of infected cells does is it contributes to this parameter, which is not any real thing, but is just keeping track of this quantity that I'm calling exhaustion level. Right? So the more infected cells you have over a long course of time, this exhaustion level will increase. And that the higher the exhaustion level is, the less effective these effector cells become. Right? They lose these functions, so they do less. So, and at some point, this all kind of levels out somewhere. And if it's going to, matching the situation that I was describing before, for chronic infection, you're going to have a chronic state where these are at some level, relatively high, a relatively high exhaustion level, and so the effectiveness or the number of these effector cells being generated will be relatively low as compared with during the acute infection. OK, so this is the basic model. So simple source and infection, right? And then that leading to, so we have some kind of saturation going on with the exhaustion term and also with the stimulation of the immune response. You notice that it's the same kind of saturation happening both with the exhaustion and with the elicitation of the effector response. So it's because it's happening presumably by the same means, the same pathway. So take a model like this. You have, and this is only a single infection, right? And you can do the analysis on this, figure out where your steady states are, your stability, all of that. And then we say, OK, well, we've understood what's happening with this model, what the situation is that leads to an exhausted chronic infection, to clearance, to an unexhausted chronic infection, which is particularly bad. So figure all of that out. And now let's add in co-infection. In the co-infection, you have singly infected and co-infected cells. We didn't include the cells that were only infected with Delta, in this case, just to keep the model a little simpler. And again, you have exhaustion level and effector T cells. And yeah. So we put this into the model. So we added a second infected class, and then basically altered all the other equations the way that you would have to make this all make sense. You have the stimulation of the effector response and the exhaustion are proportional to the sum of the number infected with B and the number that are co-infected, but less stimulus coming from the co-infected cells because of the suppression of HBV replication that's happening in those co-infected cells. Right? So there's a proportionally less amount, and that appears in the same way in all of these places. So, okay, non-dimensionalize, get a simpler version. So, okay, yeah, if you assume that there's no second infection, then it just reduces to the original model, right? So that's all simple. Steady states, nice, a fun algebra going on there. Nothing deep, just slightly annoying. And various conditions that have to be met in order to have an exhausted chronic infection. And basically, it comes down to a graph. You have to have this blue curve intersect with this green line, and the intersections have to have happen somewhere to the left of the red line. Okay, so what we have here are three possible steady states. One where all the infection goes away, one that's unstable where not much happens. Right? And you never see this one. This is just basically a break point between the two different long-term solutions and something that's bistable. So a stable solution where you have clearance, and then another stable solution where you have a chronic infection with an exhausted immune reaction to that infection, and then some kind of separatrix between these in the larger dimensional parameters, or phase plane space between them. Right? Okay, so in this extra model, in the model with the added equation for the co-infection, you get all the same equilibrium as before, and then you have a few other steady states. But it turns out that a lot of the values, I mean, subscript city here, there's a ton of things going on here. But it turns out that a lot of the values that appear in these steady states are in fact the same as some of the steady state values that occurred before, which is part of the conclusion. The conclusion will be this model doesn't tell us anything. Sorry, did give away the punchline there, such as it is. Yeah, so two of these entries are the same as in the HBV only model. So why is this, and what does it tell us about whether this recovery from exhaustion is an explanation of the long-term damage that happens in the co-infection? Well, you can do a nice little change of variables here, right? You just say, I'm going to take this B plus PD, this weighted average that I was taking of the amount of antigen presentation that's happening from the co-infected and the singly infected cells, and I'm going to call that Z. I'm going to make some simple assumptions like that the death rates of these cells are about the same. Yeah, so those are all pretty normal. Make the substitution, and when you do that, you get the original set of equations back again. Great. So what have we learned? Well, you run your simulations, and you have, okay, everything's going along here, and then I'm going to add in the D infection at this point, just kind of artificially throw some in. So of course, the number of D infected, so co-infected cells goes up. But everything else, the exhaustion level, the number of effectors actually goes down, right? And but the exhaustion level stays the same. Very small changes in the exhaustion level. So it's not the case, at least in this simple model, that adding in the extra infection can cause a recovery from exhaustion of the initial immune reaction, leading then to more intense damage in the co-infection case. So this is what happens with the exhaustion level, and it changes on a very, very, very small scale. Very short transient, and then it's done. It's just back to where it was before. So when HDV is added to the chronic HBV infection system with immune exhaustion, the equilibrium exhaustion level doesn't change, or transiently changes a very small amount. The number of uninfected cells also doesn't change. The number of effector cells is either the same or actually decreases, which is the opposite of what you would need to explain increased damage. So recovery from exhaustion doesn't explain the exacerbation of symptoms. What does happen, though, is that this weighted average of B plus PD is equal to the value of B from the single infection system. So the equilibrium level of this weighted average is the same as the equilibrium level of infected cells from before. So the total number of infected cells, B plus D, goes up. All right? If this weighted average is going to be equal to the amount from before, the B plus 1D has to be bigger than what the equilibrium level had been before. So the total number of infected cells is larger. So one possible explanation of what's going on is that it could be that this extra damage, this increase of symptoms that we're seeing in the co-infection is caused not by the specific immune response recovering from exhaustion, but rather from just more cells being infected in the system. And therefore, you might suspect that it would be just a general inflammatory response sustained over a long period of time, which would lead to this. And having an increased inflammatory response due to an increased number of infected cells could be explained. So that's maybe saying too much, but at least you could say that there might be an explanation that's non-specific as far as the adaptive immune response goes, that it's a non-specific immune response that's causing the damage in the co-infection case. And yeah, so general inflammatory response, something innate, might explain what's going on here. All right, so there are a lot of different definite improvements that can be made here. This is a pretty basic model, very, what's the word I'm looking for? No, I can't remember it. The opposite of mechanistic, phenomenological, right? It's just basically saying this kind of thing happens, and it's not really, the exhaustion level. I'm not giving a mechanism for how that exhaustion is occurring, what it is that's going on in those cells. So there could be something nonlinear, something complicated going on that explains it. But it turns out that this reaction, the results that I'm presenting here, are independent of several of the assumptions that the model makes. For example, the proliferation mechanism that you choose for the uninfected cells, the mode of infection, whether it's mass action or something more highly nonlinear that is dealing with infection. So there are a lot of things that it's independent of the specific form of the model, right, which is pretty easy to show. So there are only a few places where you could intervene in this model and change things and make them more complicated that could actually change the overall outcome in the case of co-infection in terms of whether you get a change in the exhaustion level happening. Right. So that's hepatitis delta. I'm going to leave hepatitis delta there. That is not what I'm working on while I'm here, although I'm still interested in that and still working on it. That's some work that I've done in the past. My main projects this year while I'm at the Institute are working on celiac disease with Jillian and with Suzanne Lanehart, trying to come up with a model for this autoimmune reaction. Not an infectious disease, right? It's a reaction to gluten proteins in wheat and other grains. Trying to understand what the immune reaction is that's happening in the gut, what cells are being recruited, trying to get all of that into a working model so that hopefully it can lead to something like better interpretation of tests. We have these gluten challenge tests that, can hopefully distinguish between the different kinds, whether you have celiac disease for real, or you have some kind of gluten intolerance or things like that. But that's all down the road. We're still at the beginning phases of trying to put together a model, understand the biology that's going on there. So it's basically an immune system model. The virology stuff is all out the window at this point, right? There's no virus involved here. And the other thing that I'm working on is some work that I've done with a colleague of mine, anthropologist at my home institution, who has collected a lot of data about a fishery in Zambia, or on the border of Zambia and Democratic Republic of Congo, which has all the symptoms of tragedy of the commons. Right? It's unregulated. Tons of people have access to it. It's on the border between two countries that don't particularly like each other. It's basically all set for everybody to exploit this resource and for it to go away. And in the past, for one kind of fish, that's happened. In the 50s, they lost this river trout, or salmon, sorry, that was very economically viable. And right. So it's this kind of situation where tragedy of the commons in principle should be happening. The resource should collapse, but it hasn't happened. It isn't happening. And my colleague, the anthropologist, is interested in how this kind of story, this mathematically based, economically based story of the tragedy of the commons is influencing policy and everyday life on the ground in this region. And my kind of role in this is to try and understand, based on the data that he's collected, can we explain what it is, what's going on here? Why is it not a tragedy of the commons? What is it that's preventing that kind of classic story from playing out in this fishery? So a little different from the hepatitis stuff. I just wanted to mention that before I conclude, and thank you all for your attention.